Right. Just go ahead and get started then. I was kind of hoping no one would come back after the break, but all right. I know. So my talk today is more of an application of how we used SOLAR as part of the Enigma DTI group, the Enhancing Neuroimaging Genetics with Meta-Analysis group. And basically how we used SOLAR Eclipse to mega and meta-analyze DTI data from over 2,000 subjects. So how did we use SOLAR Eclipse? Well, we used it for joint analysis, like I mentioned, to improve power for genetic studies. We created a pedigree matrix for mega-analysis, and Peter covered this a little bit, but I'll show you a specific example. We performed meta-analysis of fixed effects meta-analysis, weighting using just the subject number and also the standard error. And then we also compared mega and meta-analytical approaches and also combined the two in various forms. And I'll show you the effects of that in 5 cohorts in the Enigma, 5 family-based cohorts in the Enigma DTI consortium. So meta-analysis has been used for a long time in genetic studies, and actually a search for meta-analysis in Nature Genetics gives you over 500 articles in the past 10 years, and that's about 20% of all their papers. And already in 2014, and this was a few days ago, you have over 1,200 Google hits for meta-analysis in human genetics. So it's a big part of genetics. So what is it exactly? Well, the definition is a quantitative statistical analysis of several separate but similar experiments or studies in order to test the pooled data for statistical significance. So it's combining a lot of different studies. And so far, it's been a GWAS meta-analysis has been important for genetic discoveries of all sorts of diseases, including cardiovascular, glaucoma, cancer, obesity, and a lot of brain-associated ones like Alzheimer's disease and schizophrenia. Meta-analysis is now also being used for imaging genetic studies. And one example of this is the Enigma Consortium, which I mentioned previously. And actually in 2012, there were 4 studies that came out in Nature Genetics using different volumes of brain phenotypes extracted from images for meta-analysis, including the hippocampal volume and also the intracranial volume. And further analysis is now being performed for looking at into all sorts of volumes of all sorts of subcortical structures. But what about the brain's white matter microstructure? That's the bundles of myelinated axons connecting all the gray matter regions. This is really important for neurodegeneration and psychiatric disorders. The microstructure depends on many complex processes in development and plasticity and is highly genetically influenced, and I'll show you exactly how much shortly. So how do we measure this? Well, we use a lot of diffusion-weighted imaging. We can use it to look at differences between groups, look at tractography, try and trace tracks between different regions. This is a case of Fragile X in kids and how they're different than healthy developing kids. We can use it to look at connections, different connections between cortical regions, or we can use it to make pretty CD covers. Diffusion-weighted imaging is MRI-based, where magnetic gradients are applied in various directions. And the simplest model for calculating the diffusion in a voxel is the tensor model. And this can be calculated with as little as 6 gradient encoding directions. Fractional anisotropy, or FA, is the most basic, is one of the most common and basic models that you can get from the diffusion tensor. And it's an estimate of the extent to which the water diffusion is directionally constrained. So with higher fiber density or increased coherence and heavier myelination, you get a higher FA, more towards 1. And if the diffusion is completely isotropic and equal in all directions, you get an FA of 0. So the white matter is rather heritable. It's been associated with a lot of risk genes, and DTI is now becoming more and more prominent in genetic studies. As I mentioned, there's a huge genetic overlap between white matter integrity and psychiatric disorders as well. Even polygenetic risk scores have been associated with FA. Now, there's a recent trend towards using DTI phenotypes in GWAS, and Peter brought up a good example of using a candidate SNP approach. But there are a few papers that have been using diffusion imaging as phenotypes for GWAS. Not much has come out of it in single cohort studies. There's been a couple using FA. And there have been some almost significant hits in candidate pathways, but nothing has reached genome-wide significance, except a paper, a recent paper, which used tractography and Hardy models, not actually the DTI model. So a multi-site DTI GWAS would be really good at this point, and it would increase the number of subjects greatly, and perhaps we can reach genome-wide significance using the most basic model of the tensor and FA. But would a tract from one study necessarily be the same as a tract from another study? and how reliable are tracks in terms of protocols and imaging resolution, and in different populations. Would we have the same FA measure in different populations of children and adults? Does that mean the same thing? And we still need to know how effective they would actually be as endophenotypes. So let's find out. Our initial goal for our Enigma DTI project was to look at heritability in a vast population of subjects to see if we can get decent estimates and if these could actually be decent endophenotypes. So we perform 2 analyses. We do a meta-analysis where we run heritability at each site independently and jointly analyze the results. We also do a mega-analysis where we combine studies and run a single analysis, and both the pedigree model and the twin sample we can do with SOLAR. So I just answered my own question. In this study, we have 5 cohorts. They're all different sizes, different family types, different age ranges. All were scanned differently, except GOBS and TAUs had the similar scanning acquisition parameters. We could see these vary from everything from field strength from 1.5 T to 4 Tesla, B values, all different. We have some more lower directional resolution images, some higher directional resolution images, but the tensor model is what you can commonly derive from all of these. And as Peter mentioned before, some of these age ranges go from 19 to 85, while others are more narrow, like 9 to 10 only in these cohorts. So how can we model this all? And we are going to use SOLAR, but we have to have a common protocol before we do anything. So the first thing is to actually create a common template. And I'm not going to get into the details, but the template was created from data from 4 sites using 400 subjects total, all healthy individuals from all sorts of age ranges. And we use FSL's track-based spatial statistics to skeletonize the FA map and get a value at each one of these skeletonized voxels. And we segment that into regions of interest and look at FA and those values. And do SOLAR statistics across all those regions and also Voxelwise. And protocols are all online. So Peter briefly went over this, where we can create pedigree files for SOLAR for all sorts of family types. For the pedigree models, where we include a family ID, which group they belong to, also who their mothers and fathers are, and so we can kind of link them all, and the kinship matrix can be created. And we can do the same thing in the twins as well. Again, we just made up IDs for their mothers and fathers, and as long as the siblings who are in the same family have the same parents, then it works. And also for twin models, you have to specify that if they're monozygotic twins, so you can make sure that the model knows that they share the identical genetic information. Now, a mega pedigree file, just kind of stack them all on top of each other. You include all the information. If one study doesn't have a particular, for example, if one study is not a pedigree study, so maybe it doesn't have a geno ID, so you can just leave that blank. And you need to add this class characteristic, and every cohort gets a separate class identification. So after we create this pedigree file, how do we do the analysis on images? There's tons of data and lots of points to analyze Voxelwise, and a lot of data that gets created. Bennett will talk about better methods to work on this, but as a first pass, what we're going to do is just basically split the images and create individual SOLAR files, and then you run SOLAR Voxelwise and combine the results. And this can all be done through this kind of messy looking thing we call a pipeline over at USC now. It looks like a kind of a spider got squashed on the screen, but it's functional, and you push a button and it does all the work for you. The first module is to create the SOLAR phenotype files. And so what happens here is that we read in all the images, and you also input a mask to tell this pipeline which voxels you're interested in looking at. Once it knows what voxels to look at and what you want to covary for and anything you want to, any images you want to filter out, it'll create the phenotype files that we discussed before with all the covariates listed in there. And every phenotype will be an individual voxel, data from an individual voxel. And you can also tell if you have a large grid, you can also split this pipeline up among many nodes. So you can tell the pipeline how many nodes you want to break the image up into. So you can parallelize this pretty fast. This is pretty much what I just explained with some examples. You have a spread. You can put a spreadsheet in here which has all the paths to the nifty or analyzed files that you want to look at. The skeletonized image, which can serve as your mask. We have created one for the Enigma consortium, and yeah, that could also be your mask. And then if your only covariates of interest are sex and age, then it will include that from whatever spreadsheet you used, and it will basically create this this phenotype file where it has your subject ID, your covariates, and information for every voxel. And every instance you call of this pipeline will loop through all the different voxels. And here, the next module is where you actually tell the pipeline which instance you're running, the covariates, and if you're interested in looking at nonlinearities in the covariates like h squared, SOLAR has commands to do that, and you can just import it in here in the exact way that you would tell SOLAR to run it. And you have options in SOLAR, whether you wanted to do a univariate. Here I'm only showing the univariate case because that's what we focused on. Whether you want to look at a single site or do mega-analysis. So you can tell the pipeline which one you're interested in here, and it'll run the appropriate commands and spit out files for each trait. So each node will get a separate folder, as Peter mentioned, with all the data for every voxel. So there would be one output file for every instance you told the pipeline to run, and within that it will have one text file with all the different voxels for that separation of the image. So it will, at the end, it will have a results file with the different heritability estimates, the standard errors, the p-values for each voxel. And this last module of the pipeline will take all those text files and combine them and give you your output image, which can be viewed with any normal nifty or analyzed viewer. And again, this is what it looks like. So like I mentioned before, we're using 5 cohorts, and they're all over the place, and the imaging parameters are kind of scary if you've done a little bit of analysis looking at the different SNRs that associate with different voxel sizes and the number of gradients. You know that this sounds just horrific. However, when we run the heritabilities at each region of interest, we find fairly similar trends for a lot of regions. For example, the body of the corpus callosum, every cohort shows a fairly high heritability.
For the fornix, for example, there's a lot of mixed heritability estimates. And for the cerebral spinal tract, some have very low estimates, or most of them are pretty low, actually. So you get a good estimate of how heritable each region is for every single site on its own. Here's a Voxelwise look at the data. And now you remember that these are all different family compositions. This one here is GWAS, which is a pedigree. And this one is a large twin data set, which may be better for picking up heritability at certain voxels, so which is why you see a lot of higher heritability regions here. When we break this down into what is actually significantly heritable in each data set, see that the GWAS data shown here is about 60% of all the voxels are significantly heritable, and this is after FDR correction. QTIM was closer to 80. However, there was one TAUS, which was 1.6% heritable. That could be due to a variety of factors, mainly probably the family distribution, in that it was kind of difficult to pick up heritable effects when you don't have a large twin data set. This was maybe only a couple hundred subjects, and not many of them were actually twins. They're just siblings. There's a few pairs of monozygotic twins, so you really can't pick up a family effect as strongly. So let's talk about the mega-analysis a bit. So we mentioned a few times already looking at this, the pedigree file with all the different classes and the different representing each cohort, and the phenotype files, where, for example, we can look at average FA along all the different cohorts. One thing we can do, which is nice, is if certain groups code things a little bit differently, we can include that also in the phenotype file. So if one group codes sex as a 1 or 2 for males and females, and the other one puts an F or an M, then they can all be combined in one pedigree file, and SOLAR can distinguish between them, as long as you have the classes correctly inputted. Also, some data sets may multiply the FA value by a large factor to be able to see these as integers, and some others use floating points, so this can also distinguish between that. And then we do the normalization that Peter talked about, where we take the 5 different data sets, and you can see how different the distribution is in each one. We regress out the covariates, and we normalize the remainders and perform a mega-analysis. So what's a meta-analysis then? Here, we calculated the heritability in each group, as I mentioned, and got the standard errors based on the individual cohorts from SOLAR. And we calculated meta-analysis 2 different ways. Once we weighted by the number of subjects in each group, and also by the standard error. The number of subjects is a little biased because with the different family structures, maybe a large cohort in a pedigree doesn't necessarily mean the same thing as the number of the same number of subjects in a well-controlled twin study. So with the standard error estimate, I just said this. Oh, sorry. We also calculate this, the weighted standard error estimates for meta-analysis. And so we can weight to get an overall standard error for each of these procedures. So we can weight the standard error also by the sample size and also by standard error for each cohort. So the overall standard error weighted by the standard errors of each cohort. There's actually, interestingly, there's no standard analysis for computing a p-value from variance components-based meta-analysis. So we talked about this for a bit and decided to come up with two different estimates for calculating p-values based on the approaches that we've discussed. One is basically this for the one we weight by the number of subjects. Take the z-scores and weight those by the number of subjects in the individual groups. And also another approach is looking at the standard error meta-analysis, weighting basically converting to z-scores and weighting by the standard error. And the second, these are based on the individual cohort values themselves. The second method just computes the z-scores based on the ratio of the meta-analyzed heritability to the meta-analyzed standard error. And we convert those to p-values to look at the significance. And here's another view of the Voxelwise results, comparing the meta-analysis weighted by the number of subjects, the meta-analysis weighted by the SC, and also the mega-analysis. You can see the meta-analysis weighted by the number of subjects shows regions that are less heritable than the others. And we can see this more in detail when we look at the regions of interest. But when we plot the voxels of one method versus the voxels in the other, and this is the heritability values that I'm plotting, you can see this plot right here is the one weighted by the number of subjects on the x-axis and the weighted by standard error on the y-axis. I'm not sure how clearly it comes out, but there's a good number of voxels that were perhaps artificially high for the meta-analysis weighted by standard error compared to both the mega-analysis, which is on the x-axis here, and also the meta-analysis weighted by the number of subjects. However, overall, these are very highly correlated with each other, between 0.87 and 0.9, so correlations are pretty strong. Using the regions that are significantly heritable, like I showed before, for the individual cohorts, now we're looking at the joint analysis. The regions of significant heritability using the n-weighted meta approach is about 10% when we use one method for calculating the p-values, and 35% when we use the other. And for the standard error weighted, it's between 35 and 50%, and the mega is about 43. So here is a plot showing the different regions of interest, which might be easier to see than the Voxelwise results, comparing the different types of meta-analysis with the mega-analysis. You can see a general trend where the n-weighted meta-analysis is almost always lower than the other two. The standard error-weighted meta-analysis is usually higher than the mega-analysis. But as I showed before, there were some artificial points of high heritability. And the mega-analysis is somewhere in between. And we can also see that in the forest plot showed here for the average FA value, so the overall FA in the whole skeleton. So how robust is all this? To test this, we just removed one site and performed all the analysis again. And we get a sort of similar trend. Each one of these groupings is the removal of a single site. So all the way towards the end, we have what each analysis looks with all 5 sites, and these 4 are what happens when we remove a single, or 5 are what happens when we remove a single site. You see the variability doesn't change much with the meta-n weighted approach, or really the mega, the green kind of stays the same, and the blue is kind of roughly around the same line. However, the SE weighted meta-analysis becomes a lot lower when we remove one of the groups, and is just fairly high otherwise. So there's a bit of variation in these different methods. This is looking at every single ROI, and you kind of see a similar trend with the n-weighted meta-analysis. Looking through these pretty quickly, you can compare them. You see the n-weighted meta-analysis generally has a much lower heritability value for each region of interest. However, all the regions are still heritable no matter which method you happen to choose. So mega-analysis is great. That's kind of what the conclusion we came across. It seems to be it seems to give you a much higher heritability estimate than the n-weighted meta-analysis, and also isn't as prone to single site differences as the standard error weighted meta-analysis. But how practical is mega-analysis? Not everyone is going to give you all their data and all the genetic data, so you can see their genes in their brain. So we performed one more analysis, and that's to see what happens if you combine the data in different ways. So if you want to mega-analyze part of your data, but then you have a couple other data sets that you don't have access to, what happens if you mega-analyze some and meta-analyze with the others? So we broke this up a bit. There's 5 different ways we could combine 4 using mega-analysis and meta-analyze the other one. And we also, to remove more from the mega-analysis, we tried it with 3 and then meta-analyzed the other 2 with another 10 combinations. Not going to show you a million more plots of these, but you'll get the idea. And to make things kind of simpler, we just stuck with the n-weighted meta-analysis and just one type of the p-value estimates. So when we mega-analyze and meta-analyze together, we actually see this really interesting trend where the cerebral spinal tract is pretty much not heritable no matter what we choose. So that is kind of interesting to point out because this is a track that we're not really interested in pursuing for our GWAS analysis because it's not giving us reliable results. It's not very stable. Heritability is all over the place in different cohorts. We might not be able to get a reliable estimate. So doing things like this, we can really see where we expect the regions to be robust and carry forward for a GWAS. And here, sorry, I didn't make this clear, but on the x-axis are the different regions, and on the y-axis are the different combinations of the data that we played with. No matter what method we choose, we still do see very highly significant regions of interest when we perform heritability analysis in over 2,000 individuals. I mean, we're seeing p-values to the negative 55, 60 almost, especially for the mega-analysis. So in summary, all the results are significant and fairly stable. Even the cerebral spinal tract is fairly stable and not very heritable. It's stable in its not heritability. Even partial mega-analysis leads to lower p-values than meta-analysis, so even using mega-analysis as much as you can give you a stronger estimate than just meta-analyzing the data alone. So, I mean, there are some questions that remain in terms of heritability. So I mean, what does it really mean to combine heritability estimates from all these different sites? And is mega-analysis the appropriate approach? Our kind of goal here is mostly to look for good endophenotypes for GWAS analysis. So if we're getting stable estimates across these cohorts, regardless of all the imaging parameters and regardless of the different populations involved, then we're actually really happy with the results. We know our methods of analyzing the data is appropriate, and we can move forward with genome-wide associations. And we can be happy that the FA results that we're getting from DTI are heritable and for the most part reliable. So now we're ready. But we do need more groups for a large joint analysis across multiple sites. So far, we have several groups interested in the combined data. It's going to be over 10,000 scans. Mega-analysis will require that some sites share data. It is more powerful, so we could expect to have more discoveries. However, for the most case, meta-analysis will be performed, and even if the data is not shared, which it hasn't been for some of the other imaging consortiums, it could still lead to important findings. Again, this is basically the Enigma DTI work that we're doing. This was all using SOLAR, especially for the mega-analysis and the individual heritability results. And we have all sorts of protocols in place, looking at quality control, creating these maps, extracting all the values we're looking for and putting it into the format that we need for running GWAS and looking at SOLAR. And this is a summary of the talk. So we looked at the joint analysis, creating pedigrees, and performing meta-analysis in two different ways and comparing the approaches with mega-analysis and also combining them in different orders. And I'd like to thank pretty much everyone in the world, but mainly Peter, Tom, Emma, Bennett, David, and Paul Thompson. And if you have questions, email us down here, netajahanshad@ini.usc.edu. Right. Thank you.
Woohoo, it didn't crash. Great. I think, are we good to go? Wonderful. I have the pleasure of giving the last little talk of this solar workshop, and it's a bit from a different perspective. I'm not a geneticist. I've done almost no genetics except with Netta and Peter and our fearless leaders here. And I'm an imager, and we acquire lots of imaging data sets and rarely think of all this genetic stuff. It's just too hard. Let's find shapes, let's find metrics, let's do a voxel-based morphometry, let's do a tensor morphometry, let's do all of these really complicated fun things in 3 and 4D, but genetics, we just can't do it. I'm here today to talk to people online, to talk to those of us who've managed to stay in this beautiful state. It's not that hard. I can do it. Actually, Mary Ellen can do it, Tricia can do it, and I can help a little bit. So, overview of what we've put together. Little motivation. You've seen a lot of this over the last 2 days, talking about a couple of different packages for heritability. Then what we do best is play with simulations. Let's start with something we know what the truth is to see if an engineer can actually run these software packages and get results that make sense. And then I'd like to give these things to you. So those of you who have your laptops, either now or at home tonight, later on the video, can go ahead and grab them all, try them out. I'll do a little live demo unless something crashes. And make this really simple to use and conclude with a few remarks. So first, motivation. So we've been talking about a lot of linkage studies and a lot of ways of looking at SNPs. let's take a step back, and from an imager perspective, we often have twins, some maybe siblings with a healthy control, and there are a lot of these studies around where we just haven't considered our ability to do a genetic component. Well, heritability is accessible. It's the first thing you'd look at in terms of genetic association, and it can lead to more robust collaborations with imagers and geneticists on the same sort of project. Let's first see if these imaging metrics have some sort of heritable component to them. Well, this has been done a lot. Starting in Paul's group in 2001, we have cortical gray matter density is heritable. Cortical area thickness is heritable. Resting state fMRI connectivity is heritable. White matter microstructure is heritable. Okay, we're done, right? No, actually, we're just getting started. We're just able to start pumping through these large imaging phenotypes, getting the data into consortiums, and looking at structure and finding is this heritable, are these quantitative phenotypes heritable? What part of these things are available? And the software, as Peter alluded to, is coming online. It's here ready now that anyone can play with and use in a scientifically justified manner. So we're ready. So we can do this with more than twins. We can do this with deep family structures. Let's see what we can do. And there's this big mystery of normal brain function is just there. Where are all the heritability components coming from? Let's see what we can find. So first of all, a couple of study designs. A lot of people have used twin studies for this. You have monozygotic twins, dizygotic twins. H squared can be computed quite simply. Problem solved. Not quite, it's a little bit more complicated than that. That's basically what we're going after. Well, twins are rare. I have 3 kids, none of them twins, thank God. But how would we analyze different family structures? Well, SOLAR has taken this and made it really simple. Peter loads these pedigree files. They're not hard to code. We just see that they're text files. We can code an entire Amish population using your online thing. I was Googling that while you were talking earlier. You can make these things from Ancestry.com if you feel like it. So how would we get these into a statistical package? Once we have these, what do you do? Well, you can't just take R squares. There's too many components, and that's what the variance components method is about. We say the total trait variation that you exhibit in a phenotype is genetic plus environmental. We break down the genetic component into additive, which are the little components we're interested in, and the dominant effect. And then we have the environmental variation, familial household plus random and individual. And we isolate 3 components, the additive effect, the common familial household effect, and the random individual variation. And we have that the trait variation is approximately the sum of these 3 things. And we define ACE as the ratio of each component to the total amount of variation. So we have the additive effect A is the sigma squared A over the sum of the other 3. C is the common, again, ratio. And E is the environmental, again, ratio. So we got it. Been talking about this for 2 days. This is the perspective we're starting at. So what kind of software is out there? Well, there are 2 major packages that our collaborators have been using and talking about. One is SOLAR. This is the SOLAR workshop. And the second is OpenMX. SOLAR is a sequential oligogenic linkage analysis routines. Here's the URL. And the paper that sort of started it all off is from 1998. Over 2 decades of work has gone into this. It's Tcl-based. Peter explained it looks a lot like SAS or any other interpreted statistical package. The second one is OpenMX. You can see from its logo it has a little bit of a less human focus. And it's really going after structural equation modeling. And it's an open source extended structural framework. It's R-based, it's open source, it's installed by the standard CRAN package mechanism, and it just appears. And it's in R. Statisticians love R. Engineers don't mind it. We kind of gravitate to MATLAB and other languages, but it's something we can deal with. So let's first go into SOLAR. It looks like this. We load a pedigree, we load some phenotypes, run a couple of commands, and stuff pops out. It's a little command interpreter that we can deal with. What are those things that go in? There's a pedigree file and a phenotype file. A pedigree file just says, who are the people? Who are their parents? What is the relationship structure? You have to go, as Peter warned, 1 level deeper than the people you're looking at. Everybody's got to have a parent. And then we have a phenotype file, and that's the stuff we're measuring. So you can have a lot of people in the pedigree that you don't care about, and just specify the observations you have. And that goes in the phenotypes. And then the outputs of SOLAR are great little commands that say your heritability is 0.48. With a p-value, blank, it's significant, you write your paper and you're done. Well, if you believe that you set it up properly, you are done. But I strongly recommend doing some test cases to make sure that you've set up your program, interpreted the file formats, and that your brain to the Solar programmer's brain are in line. Because there are lots of ways of making human and human-computer errors in between there, where everyone's doing something reasonable, but something unreasonable happens because of sort of communication issues. Well, OpenMX looks similar. You can run R in many different ways. This is running in Windows, and I have a Mac, and I'm going to run it in Linux in a bit. R works great. Notice the version here is not the newest as of January 21st. OpenMX likes 2.15, not 3. I'll say this a couple times. Don't get 3. It'll make your life miserable. So we have a little bit of an old version of R. You type require OpenMX. That becomes OpenMX now. Now, rather than going into all these external files, things are just variables. So it's your choice how to get them in here. Well, except for the structural equation modeling. OpenMX is a structural equation modeling language. You need to specify the structural equation models, which means programming a little bit in R to specify them. Twins are not hard. It's about 15 lines of code that you can snip off 100 different web forums. Non-twins are very complicated and interesting and involved, and so I'll just go to say OpenMX doesn't readily support non-twins unless you're going to take a huge amount of effort. The phenotypes just show up, and you can use whatever mechanism you would like to load the phenotype into a R variable. And then the outputs are R variables, everything nicely loaded into memory, and you can save them in any way you like. R will save them in the .r data file, or you can print them into text files. Anything works. It's both a blessing and a curse. It's not nicely formatted, but they're there. So, a little bit of a breakdown in the terms of the comparison. Solar is currently closed source, black box. Peter and John Blagera are interested in collaborating, but it's not something, it's just there. It's much easier to specify complicated pedigrees in Solar. It's load pedigree in one of many file formats. It's single-threaded execution. You can break it apart manually, much as Seta and we have done a little bit, Neta and I have done a little bit. And it's multi-platform Tcl-based. So if you're a Tcl hacker, of which I doubt there are any in the room, it's your dream. Otherwise, it's not bad. OpenMX is difficult to extend for complex families. It's possible some people have hacked it. It's just not a really pleasant experience. It requires a little bit of tuning for the maximum likelihood estimations in terms of the ACE, and it tends to have some conversions issues that you have to retune these things a little bit more often than Solar. But it integrates with R's cluster processing. If you have R installed on a cluster and have installed the R compiled libraries, it farms out without too much stress. And it has a well-managed active forum with lots and lots of help documents and streams of people arguing about what the best way to do something is. So those are the packages we're thinking about simulations. When I came to this project, Bennett, why do you want to simulate? It's crazy. We have 10,000 data sets, just play with the empirical data, reproduce what we've done. Well, I'm an engineer, and I'm always worried that we're doing something wrong. Usually it's true. So the key reasons are first to debug, making sure that Solar's mind, to my keyboard, to everyone who's working on the project is self-consistent. 2 is to assess performance. If we're going to know how these methods are, we need some ground truth, and we don't have ground truth on empirical data. And the 3rd is to wrap up this method comparison. So debugging, there's how do we know if the software is installed, configured, the libraries on my different machines may be different. Am I using things wrong? Am I calling the wrong command? Did I read the wrong documentation file? If we have a simulation and you can run it quickly and cheaply, you can confirm that you are properly thinking about the problem. And also, if we're going to send stuff to Texas for help, or Maryland or someplace else for help, if I have a simulated data set, the IRB doesn't care if I share it. IOCOC doesn't care. No one cares. Yes, it's great. We can give little simulations and just share them all around, isolate problems. It helps our lives a lot if you guys can play in a simulation that's just fast and quick and isolate whatever problems you're having. Second is performance. A lot of these things we're doing, and we've been talking about over the last 2 days, are edge cases. It's not something that Solar was designed to do. Yes, it can do it. Of course it can do it, but it's not something that's been heavily tested. So there may be coding bugs, theoretical bugs, thought bugs. The user manual may have been vague, and you could just be misreading it. Something could crash, the file format thing. They didn't expect to open up 1 million files, and when you do that, it kind of deleted your hard disk. Problem fixed. So we need to look at all these edge cases and where we know what the truth should be. And then a generative model is really nice for producing power curves that statisticians love. Grant reviewers love this little power curve. Well, you can do all the simulation framework I'm going to go into and that Mary Ellen and Tricia and I have been doing, but Solar has 2 great commands for making power curves. H2 power, heritability power. You load up your pedigree, and it makes your curves for you. It's beautiful. It's much, much faster than doing all this external simulation stuff, and it does it all internally. But the internal one doesn't solve all of the other problems of debugging and conceptualization. And for those of you reading later, there's also another command called power, which is more general than the heritability. It does the whole QTL type stuff. So if you like power, H2 power, those are empirical. There's also a paper that came out in 2013, and I believe October, that has really concise power estimates on theoretical power models for all these maximum likelihood tests. So there are theoretical underpinnings if you don't want to go to Monte Carlo. Just figured I need to point that out here. Third, why simulate? They provide a virtual, scalable access to a lot of people. Want to try it on 5 people? If we want to try it on 500,000 people, it's just CPU time, and now that's really cheap, as opposed to, Netta, can you grab a couple more people in this consortium, please? You know, it doesn't work that way. It's much easier to grab a couple more subjects. We can simulate class differences. We can introduce all sorts of artifacts and processing problems and make sure that things keep working. And once we have the results, we can know if they work by meaning what's the bias of our estimator and what's the precision of our estimator. We can compute that exactly and analytically, and we got it. No problem. Remember, mean squared error is both the bias and the variance. Without a truth, you can only compute the variance. If you do resampling, well, you're missing the bias. If there's a fundamental flaw in what's going on, you're not going to catch it if you don't have the truth. So, taking a step back, what are we going to do in simulation? We're going to look at a bunch of different family structures. And the way we broke it up is, how do things work on twin studies? So monozygotic and dizygotic twins. How do things work for nuclear families, where we have a pair of non-twin siblings plus their parents? And how do things work in grand nuclear families, where we have a pair of siblings, their parents, and their parents? Just to sort of say, does Solar make sense? If I'm an imager and I'm collecting images on populations, how deep in the family structure do I need to go? How many people do I need to collect? Is this even going to be possible with my traditional study on disease X? So what we did is we took these twin structures, the monozygotic, the dizygotic, the nuclear family, and the grand nuclear family, and we swept pedigrees. So we just randomly created pedigrees between 8 and 600 subjects. And then we simulated the additive effect. So we swept this between 0 and 0.95 of the total variance, so no additive effect to almost all. And then we swept C. Well, keep in mind that A can't exist if A plus C is greater than 1. So we went C is 0, 0.3, 0.5, 0.7. But if C is 0.7, A can't be bigger than 0.3. Those will show some lines in the curves where we just say, can't simulate here, it doesn't exist. And finally, we'll create 100 phenotype files. We got all of this stuff. We just use the generative model of the multivariable norm. So the output is X beta. We randomly generate an age for each of these people. We have a random beta, which is the interaction term. We the fixed effect. We add multivariate random noise. In MATLAB, MVRAND. In R, it's RAND, MVN, something like that. Y is a simulated phenotype. X is a simulated covariate matrix. We just used age in all the simulations I'm going to show you. If you want to tinker with our simulations, it's easy to add more. B is an arbitrary coefficient, just to say that there is some fixed effect. N is a multivariate normal, which is ACE, the specified looping structure. 5 Phi is the kinship matrix, where monozygotic are half, dizygotic are a quarter, parent-child a quarter, et cetera, et cetera. And gamma is the assumed common environment between the relationships. So if you're in the same household, it's 1, and it's 0 if you're not growing up in the same household. So we did this, and here's an example of what happens. The covariance matrix is ACE between themselves, so it's degree of variance, and A and C. The guys that got twins the other way. So now that we've created all these files, you can imagine it's a lot of files. We write them all to disk, it's all there. We run these in 2 different packages. So let's look at the twin study first and have a little bit of a throwdown, what's going on. We did what we just said, just twins, because OpenMX doesn't really support the complex pedigrees very well. We have to hand code a lot of different things. And we're going to show a number of results that look like this. So for each set of experiments, each level of the simulation, I'm going to put up a little heat map. And on the x-axis, it's the number of subjects. It's not linear here, 8 on the left, 600 on the right, it's going up. And on the y, it's the simulated heritability. 0 at the top, 0.95 at the bottom, and if C is greater than 0, the bottom part of it's going to be cut off. You can't simulate A if A plus C is greater than 1. And we're going to color code this so that it goes between 0 and 1 in a nice little heat map. Non-heritable stuff is red, heritable stuff is yellow. So first thing, twin study, cross-platform comparison. We have C at 0. This looks really nice, red, not really happening. This should be bright yellow, right, 0.95. That should be red because it's 0, and then it should be in a linear gradient. With 8 subjects, both things are having problems. With 600 subjects, both look nice linear ramp as best as we can tell. And from the back of the room, things look like they're doing pretty well. Let's look at the bias. So what we're going to do is take this value, the true value, and subtract it from each row. So how wrong are they in absolute value? In not absolute value. So you can see that we are low on heritability with only 8 subjects, and then less than 0.1 everywhere else. It's black in terms of this color scale that we can see on the monitors here. And the same thing over here. OpenMX has a little bit more problems. There's some color that I can see all the way out. That means the bias is up here, sort of for quite a few subjects. This is at h squared. So this is h squared estimated minus h squared true. Where does the percent? Yeah, that's fraction. So there's a 95 is the fraction of 100. Thank you. So if we look at the difference between those, OpenMX is more biased everywhere except very low heritabilities with very few subjects. Woohoo, Solar wins. Great thing we're in this workshop. I still get to keep my job. So putting that all together on one slide so you can't read any of the axes. We have, from back of the room, they look good. The bias is higher for OpenMX, and the variance is higher if we look at the standard deviation of those simulations. Great. Solar is working better for the twin studies and quite a bit better across the board. Okay, that was C of 0. Now C of 0.3 we're having a little bit more trouble with, and there's probably a communication error between sets of computers in my brain and how we're simulating everything. So we're debugging this problem, but this is where we are today, and this is the code that you can run. The heritability is high for Solar, and more appropriately linear ramp for OpenMX. And if we plot this, we're twins with a 0.3. Solar is biased. OpenMX looks sort of like with C of 0. We ramp up C again to 0.5. Solar is having problems. 0.1, Solar is having problems. So if we put it all together, no common effect. Solar is winning by a lot. Making sense. It's a really stable method. Something is happening with the higher common effects that we are working on debugging. And if you have common effects, please talk to us, and we'll resolve this definitely by next year. But that's why we have these workshops, because we've been working on email for a while now. So great. What about more complicated things? Let's take the nuclear families and the grand nuclear families. And if we do that same thing, 8 to 600 subjects, C of 0, looks beautiful across the board. We have a nice linear ramp, a nice linear ramp, a nice linear ramp. These are all 8 to 600 subjects. So the axes agree even though they're not linear. There's fewer twins and more twin families, fewer grand nuclear families with the same number of people shown on this page. And for the same number of people, the bias is high for about 8, sometimes 8 to 20, 8 to 20, a little lower bias, but progresses a little more. The variance, the same sort of deal, 8 to 20 sort of has problems. Once you get to 40 to 80 subjects in sort of any of these study designs, things are really working. Great. Modern imaging studies often have 40 to 80 subjects, even if they weren't planned to be genetic. That means I can go to my collaborators in psychiatry and say, hey, you were using siblings as controls. Why don't we fold on a secondary analysis of heritability? Maybe that'll pop up something that's really interesting for you in terms of genetics. Maybe we can go to the people looking at all these different diseases and throw them on, because those study designs are really feasible for anyone collecting weird family structures. And SOLAR doesn't care if you do siblings in one and cousins in another. We sort of lose power when we go to more and more distant relationships, but we can fold them in. Now, in twins, we have this problem, but hey, look, it's working really well. So if C is greater than 0, for nuclear and grand nuclear families, we're not having this problem. It's working great. There isn't this glitch in terms of SOLAR having biased estimates. So we're really happy with those. If we raise it higher, no problems, no very low bias, very low variance, and again, with very high common effect, no problems. So it's really working well. We have one more bug to fix. So as of now, our recommendations are if you don't know the common environment and you have a complicated study design, SOLAR is really awesome, or come work with us and we'll see if we can fix whatever is going on. Otherwise, OpenMX is easy to code up and it's working. If you know that C is 0, SOLAR is giving better answers. And if you know that C is greater than 0, nuclear, grand nuclear, complex study design in SOLAR, or the OpenMX twins. So, a little bit of a live demo to show you what I've been doing, and we'll see if this works pretty well. So, go over here, duplicated. Log out. So this is a virtual machine that I have posted to Nitric. You can download this hard drive and fire it up yourself with all of the warts and problems. It's licensed to the Bennett Lehman user. If you'd like to get your own SOLAR code, email the SOLAR registration people and you can change your name. And the password is SOLAR 2014. That's also on the hard drive, all lowercase. Great, it fires up a Scientific Linux 6.4. Works great. It's basically a CentOS clone. This is what our genetics people use, so that's what I installed this on. I like using RStudio. It makes things nice. Close everything out. So here is R. This is R 2.15. On the hard drive, on the desktop, there's a live demo. And we've structured this into 3 sets of scripts. The first script runs all the simulations through OpenMX and generates a SOLAR file to run in whatever grid engine that you want, the text file that's all the tickle commands lined up. And then 2 ones that'll make the figures that you saw on our posters out there and all the posters in this talk that you can just generate yourself. You can see precisely what we did. So this script is run first. It simulates monozygotic and dizygotic twins, require OpenMX. These are the packages that need to be installed, and the instruction manual we posted online says how to get these and how to get them into R if you aren't an R fan. We set up the output directories of where things go, and so this goes into a folder called Live Demo with Twins to be very cryptically named. Makes all the directories, and then we set up how many pairs we want, the number of pairs, set this seed for the MVRAN. This is the so that if you run this every single time, you'll get the same random numbers. If you want to change it, go ahead and change the seed, but for reproducibility, setting your random number generator is a very good idea. And then standard deviation attribute, C-squared attribute, this is what we're going to loop over. And then we loop over all of those pairs and the twins that I just showed you. We write the commands out to a file to be run in Solar, just the load pedigree, load phenotype, all of the things that need to be done that Peter and Netta showed earlier. It's going to generate for you, and it's just on and on. If you want to go off the edge of the screen, everything that needs to be written out. And then we're going to create each of the phenotype files, looping over each out of genetic standard deviation, and figure out what they are, create the file, write it out, create the file, write it out, create the file, write it out. So we haven't integrated all of the solar eclipse yet. We look to be doing that in the next 6 months or so that we can write out an imaging-based volume, and all of this would be a true imaging-based simulation. For everything we've been doing now, we've been breaking down the images into voxels and farming them through the clusters that way. So we create a file to output the solar beta estimate so that every experiment gets written to a separate output file, and then it loops over every Monte Carlo iteration, and on and on and on. And it's not that complicated. All it's doing is looping over the model in R, writing out a file. Looping over the model in R, writing out a file. This is where we specify the MX model. This is the magic code that you can steal that gets a twins model for OpenMX, where you put what the A, C, and E are. Then we run the model with MX run, save the MX results, write them all out to disk. So this should take, hopefully, seconds. So it's running, it generated all the files, now it's running them. Done, almost. There we go, and that's just some warnings that the convergence was possibly non-optimal. If you read on the R forums, it dumps that pretty much all the time. So if we look at the output directory, /data, cd solar, live demo, twins, twins 4. So it just created a set of files that load pedigree blank, load phenotypes blank, model new, trait kindergen, which is the trait that we made, covariate age, out directory, so make sure you write it there, run polygenetic. Rinse, wash, repeat, rinse, wash, repeat. And that's all it does. If we go solar, MZ twins, dumping all of this, you can output this to a file if you want, but it's running all of the heritability estimates, and it's done. This looks like the slide I showed earlier. And then if we open up the pull solar and run this, this file loads all of those individual files in, makes an R data structure, and plots the heat map. So if you want to generate every figure I've shown, just run the ones in the other directory, and I'll show you those in a minute. And the last one, see those heat maps that just got created. So desktop, live demo, pull OpenMX, and hit the run button. Does the same thing. And we generated link to live demo output, twins OpenMX bias, ta-da. Only 2 pixels, took, I guess, 10 minutes for us to walk through to see what this is like. We wait about, with a single thread, 2 days to finish all the simulations, 2 or 3 days, depending on your CPU, to finish all the simulations that we did earlier. Or you could parallelize it, and it just takes up 1 CPU. One thing I wanted to note is if you're going to do this, it's a lot of little files. All the simulations that we have take about 27 gigabytes of uncompressed data in terms of just the output. No big deal, everyone's got that space. The issue is modern file systems have this thing called inodes, and there's one inode per file. So if you used a regular 30 gigabyte disk like this one, you could only store about 2 million files on 26 gigabytes, and I think that's enough. Well, if you're going to generate a bejeebers number of small files because you're running all your imaging through, you can't use a regular file system. Fortunately, Linux and all these others come with modern file systems like ZFS that can do this, and I have a little tutorial on how to set up your file system so it's fast and cheap, and you don't run into inode problems, and you can see that there are an order of magnitude more inodes on data, even though it's essentially the same size of disk, and we don't run out of inodes. That was a learning curve. So great, not so hard. All it is running a couple of commands, and we have these simulations that will generate an ACE model with any random effects for you to look at. On the virtual hard disk, there's a file called rscripts for ACE twins, AE ACE nuclear family, and ACE twins solar MX. So AE, ACE, and this is both solar with the nuclear and grand nuclear, and the same set of scripts are in here to run all the simulations that we looked at today. This is the code that generated the figures. I think that's what I wanted to say on that one. Let's see if this can come back here with Shift F5. So that was Scientific Linux 6.4, 64-bit OS, Solar 7.3.2, which is December 31st, R215, I'll say it again, not version 3, and OpenMX 1.3.10.12. That all works. How long did this take? The twin study took 155 minutes in OpenMX versus 266 in Solar. A lot of the Solar is file I/O, and OpenMX, a lot of that was file I/O. So OpenMX is actually quite a bit faster than Solar, but if you're going to be waiting, you might as well wait for good stuff. The ACE twins took 242, Solar 382, so again, longer. The nuclear families took 600 minutes, and the grand nuclear families took 1,800 minutes. So the additional pedigree structure takes longer for the same number of people in those families being simulated, but it's not blowing up exponentially. But a lot of that's file I/O that we hope to get resolved by integrating Solar Eclipse. And those times are on this laptop. So you can get all of this on our Nitrix site. If you click on Docs, IIG's 2014 demo, the sample scripts are there, the virtual hard disk link is there, and setting up Solar and OpenMX on Scientific Linux, it's a 40-point step-by-step graphical how to configure your own Linux workstation with everything I installed. I took a lot of screenshots. So almost ready to wrap up here. Does any of this matter? The H-squares that we were finding in the white matter tracks were about 0.5 arguably. About that ballpark, plus or minus about 0.1. So if we look at this cross-platform twin study, and that had a C effect of 0, that would be about here. Okay, that doesn't look so bad, but if we put that onto the bias map, that is about there. And so the difference between them in terms of bias is on the order of 0.3, some maybe 0.4, somewhere in there. That's not a trivial amount of difference in bias between Solar and OpenMX in that approximate range with approximately 400 subjects. So this could be a problem, which is a little bit surprising. Those looked really good on the nice flattened out color maps where we were looking on a sort of crummy projector. But when you drill down into what is the quantitative difference, it goes away if we have lots and lots of 10,000 subjects. But in the imaging big world, hundreds, these differences really still exist. So we went and ran this in Solar and OpenMX on the QTIM study, just the twin pairs. QTIM had a bunch of other people in there that we excluded, so this is less than what Netto was showing by quite a bit. But we have heritability on 0.5-ish, 0.7, 0.8, pretty high on this slice of the white matter. If we go to OpenMX, we look at that scale, it's down at 0.01. We wouldn't even see anything if I didn't jack it up a bit. That's quite a bit worrying. And if we go up a slice, the same sort of pattern continues with about 400 people and this imaging data, not everything, but this imaging data, With this level of environmental variance, OpenMX wasn't finding the heritability, but SOLAR was. And that's confirming that that is corresponding to the simulations that say there's about this degree of bias. So it makes us believe our simulations a little bit more, that we're not simulating whole cloth completely, that these have basically the characteristics of this imaging data. That's why we picked all the numbers that we had in our simulations. And it says that SOLAR is working pretty well. So this common effect thing, we're kind of cruising along with the C of effectively 0 in the white matter tracks from Peter's study from a couple years ago. Wow, 4 years ago. Feels like yesterday. So we're okay. It's going well. In cortical thickness, it's been reported to be quite low, less than 0.1, less than 0.05 in cortical thickness and brain volume, and slightly higher possibly in the white matter of neonates. So this is something that we'll probably need to deal with, but it's not a thing that's popped up recently. This mode hasn't been really hardened, and that's sort of our job here, to help you guys find us how we can harden the program and make this really easy for everyone to use. So the overall e-science objectives of this is in the title of our partnership, is we're providing a freely available platform for learning, and we're not really adding much value except in terms of the hardening and making the instructions accessible to regular old engineers, people who haven't had a strong genetics training, who want to get into it, trying to bring people who have familial structures in their imaging studies, and making this just stupidly easy to use. Oh, of course we'd put on SOLAR and see if there's a heritability and see if the geneticists really want to be involved. We're working on quantifying the differences between the OpenMX and SOLAR packages. There's a lot of energy in these communities and pride in terms of the tools. And we're trying to give a systematic comparison so the communities can talk to each other. We're not really vested in one or another other than we're on the SOLAR team. But we want to see the best science get done and help it get done easily for the types of models that matter to the imagers that have not deeply considered genetic linkages and aren't doing GWAS. And we're working on illustrating analysis with other environments. Our statisticians really like R, and they want to be able to fold stuff in easily with R. So we've released these scripts that show how to run them in R in a sort of way that our statisticians like. We're working on integrating with more advanced packages. Netta has the Lonnie packages set up, but we're looking for what people want and what our users make. So we'll be continuing expanding on this in the next year, integrating with Solia's Nifty and Jifty support and the pipelining platforms. So thank you. Netta has been invaluable in getting this done, as well as Peter and Tom and Paul and John and David, sort of like the core of who is making this happen, and the entire Enigma DTI group for sort of initiating all of this. We would not have started this massive effort if these data sets were not available and we didn't have to face these deep problems. And believe it or not, the NIH and IBIB supports us, and we are incredibly grateful for continued funding and the rest of my lab who's put up with this. So thank you. I'm happy to take questions or turn it over to Tom. Wonderful. 10-minute break. This is yours. So we're trying to drum up business and make it clear, right? So thank you. I can present David's slides.